And our next section of the program will be commenced with Diwakar Devar talking on the role of diet and gut microbiota in determining response to immunotherapy in melanoma. Welcome.
All right, people can hear me, right? Good. Disclosures. All right, so the whole idea is for this to be somewhat interactive. So if you have questions, if the people on Zoom have questions, you're welcome to interrupt and ask. And I am happy to take them at really any time. So kind of why we have started focusing on this area is really because of all the stuff you've already heard, right? So the point is that in this disease, we're now curing about 40% of our patients, which is great. It's outstanding. Like 40% of patients with advanced cancer do not need to worry about their cancer coming back after we put it into a long-term remission. Our only problem is that, one, we don't know who those people are. That's why you heard the talk about biomarkers. And two, we don't quite know how to make it better. And so we've got these new treatments. TIL, for example, has just been FDA approved. And presumably those patients will do well long-term. Certainly that's the data from the Steve Rosenberg lab. But what we are seeing is an inflection point for the first time in a long time where our patients are durably surviving. The question is, how can we make it better, and how do we know this is to be the case? So the first and the most interesting thing I want to show you guys is actually data from the public domain, right? So how do you know that we're doing a good job? We know we're doing a good job because for the first time in recorded history, the national death rate for melanoma is going down, which means that unlike every other cancer, right? For example, uterine cancer or non-melanoma skin cancer, where the death rates are going up, in melanoma, the death rate is going down. And the only reason of going down is certainly there's some targeted therapies that are helping putting people into long-term remissions, but it's also predominantly the immune therapies that are really doing a lot of that work. And so as we start to think about why this might be the case, we really have historically focused on three things. You know, you look at the cancer, then you look at the people who get the cancer, but nobody's really looked at the stuff inside the people that get cancer. And that's kind of what our group is focused on. So the question really is, why do we do that? And the answer is very simple. It's a math problem. All right, it's a math problem. So the human genome, right, all 23 copies of you and what I and all of us have, contain about 24,000 genes. Well, it turns out you need about 30,000 in order to survive. So where does the 6,000 come from? Well, as it turns out, you can't walk around with it in your wallet. And it happens to be in the gut bacteria that you have. And really what this is, it's a symbiotic relationship. We have them, they have us. They feed literally kind of like your 20-year-old kid in your basement. They feed off you. And what we do is we need them in order to be able to use that bacteria for certain things like vitamin K. We don't produce vitamin K. Humans need vitamin K. Where do you get your vitamin K? From the bacteria. So as it turns out, we've been using them for a whole host of things that we've known about. But what we're now realizing is that they may actually help immune therapy work a little better. And how that happens and why it happens is what is the focus of our group. So really sort of four parts to this, and it's really meant to be very simple. And I've broken it down into four things. So the first thing is, what can you take in that affects how these drugs, the drugs that we're giving you, work? Two, what are those bugs and how do they make things work or not work? Three, can you try to change that? And four, what do you want to remember if you don't remember anything else of what I've said? So simple stuff, antibiotics, bad, right? So why do we know that antibiotics are bad? Well, we've tried it in mice. Now, again, if you do mouse work, you know that you can make almost any tumor smaller in a mouse. You just need to squeeze it hard enough. But people have shown that in mice that get, in a cancer model of mice, that the receipt of antibiotics can depress the effect of immune therapy. Then humans, studying other humans, have done meta-analyses that have shown that when you look at the recent receipt of what are known as broad spectrum, so not narrow spectrum, but broad spectrum antibiotics, we reduce the likelihood of people developing responses to cancer and people having poorer cancer outcomes, especially if they're treated with immune therapy. And certainly this has now been also shown for CAR T therapy, that is cellular therapy, in data from the Penn group. Macronutrients, you heard here earlier from Dr. Najjar about the study in MD Anderson. So they have shown that, for example, there's a certain inflection point when it comes to fiber. And certainly when you give people a permissive amount of fiber, what is thought to be about 20 grams a day, you certainly have slightly better outcomes in patients who receive immune therapy compared to not getting that minimum amount of fiber. Certain people looking at in a healthy donor patient setting from a group in Stanford showed that fermented foods, so not necessarily just high fiber, but foods that are rich in fermented products, can also have non-overlapping positive changes on the gut microbiome as well as the host immune system. And the key thing to keep in mind here is that fermented foods in this context are typically things like sauerkraut and kimchi, not beer, right? So it doesn't mean to say that if you go get a keg, you're going to do better than the guy who didn't drink a keg. But what we're looking at is really the intake of high fermented foods at a sufficient quantity for a sufficient period of time. And in addition to that, increasingly, we've got data that is looking at non-nutritive sweeteners. And non-nutritive sweeteners are all the stuff that is out there, right, on the extreme right of the coffee thing, where it's things like Splenda, Sweet and Low, NutriCal, NutriSweet, Equal, all the stuff that we've been taking ourselves and telling ourselves that it's better to take than sugar. Well, it turns out it's actually quite bad. And this was certainly shown in a very elegant preclinical paper from the Crick Institute. So you can see here that mice that get high amounts of non-nutritive sweeteners do a lot worse. But we try to do a simple experiment here, right? So we said, you know what, the FDA must know what the right limit is. How can they possibly not know? So it turns out the FDA limit of sucralose, aspartame, acesulfame, and saccharin are all listed there. But let's just talk about the sucralose daily limit for a minute. So sucralose is a sweetener that is typically found in Mountain Dew. I use the term Mountain Dew because I used to drink Diet Mountain Dew, and I know it gives you a great rush, which is why it's fantastic. And sucralose, the daily limit is about 5 milligrams per day. And so part of the reason why we studied it was because it was one of the drugs that the FDA thought actually had a very low limit that was considered to be safe in humans. So you can see aspartame 50, sucralose 5, right? So we said, you know what, it can't possibly be that bad. So let's just look at how people do. So we'll take the amount of sucralose people are taking in. And so the way we calculate this is I've got a couple of people that work for me, for my patients and other patients that have been seen by Dr. Najjar, Dr. Kirkwood. These are the little people that come up to you and they ask you these annoying questions and they keep persisting and they keep asking you stuff until you give them an answer. And they hunt you down in the parking lot. And then once you give us that answer, we then take that information, we put it in a computer and we separate it by your weight and we come up with a number. And then we see how people have done, right? So what I've shown you here is the daily limit. So the sucralose daily limit is there on the right. So the first thing you see is that the vast majority of the people that are telling us how much they take are not eating very much, right? The daily limit is 5 milligrams per day. The vast majority of what people are taking is about 0.2 of that. So the vast majority of everybody that we ask this question to are taking about 10% of what is considered the daily limit. And when you use that as a cutoff to look at how people have done over time, firstly, you realize that it actually affects how much you do quite a lot. So when you look at the people who are taking just 10% of the daily limit, again, the daily limit is 5 milligrams, and we use a cutoff of 0.2, so that's approximately 4% of the daily limit. When you looked at 10%, it was poor. When you looked at 4%, the difference was quite stark. And this is actually suggesting that if you take sucralose at a certain dosage, you can actually negate the effect of immunotherapy, but quite a bit. And the way you can think of that separation of the curve is that separation of the curve is exactly what CTLA-4 does to checkpoint, right? So when you take a CTLA-4 inhibitor, you improve your likelihood of responding by about a hazard of 0.7. Taking artificial sweeteners just negates that, right? So artificial sweeteners, probably pretty bad. This data is actually, we've now replicated this in a mouse model, and we think it's probably to do with a certain key amino acid known as citrulline and arginine, and this work has currently been submitted to a journal. So what else, right? So if the stuff that you take can affect the bugs that you have, then what bugs are good or what bugs are bad? So as it turns out, you know that people who do well, we've shown, other people have shown, that people who do well and people who don't do well have very different bugs. And these bugs are quite interesting. So again, most people here don't really care about bugs, but I care about bugs. But most people won't remember that I care about bugs. But the short answer is there are a whole bunch of different bugs that are very, very, very interesting, right? These are all these different bugs, and you see a lot of them. Most people think of things like the stuff that's in the back of, the yogurt. And certainly those bugs tend to be good to some extent, but there are lots of bugs that you may have heard about that are in these probiotics that you take that actually show up in the stuff that's bad. And the short answer is that we think that you can essentially develop a signature of what's good or what's bad, or who's going to do well, who's not going to do well. And I'll show you some data about that. But the key point is that reliably, multiple people here, Europe, MD Anderson, Chicago, Montreal in Canada have shown, actually even the Koreans have shown, that the bugs of people who do well across a variety of cancers treated with immune therapy are very different from the bugs of the people that don't do well. And the question is why, right? What is it about these bugs that do that? Now, it really turns out that what the bugs do, if you happen, for no reason that you necessarily can control, if you happen to have a series of adverse organisms, right? It's kind of like you can't control who to a large extent, you can't control the bugs that you have. It's kind of like saying, I kind of wish I wasn't married. Or my wife's second cousin wasn't this crazy person, because again, you like your wife and you got married to the person, but she's got an idiot cousin. You can't do anything about that. So the bugs in many ways are like that. You have very little control over the things that show up. But those bugs can, if they happen, right? Again, cousins being cousins, if they happen to be bad, they can exert a very deleterious effect. And the reason they exert the deleterious effect is because they produce and they engage in a crosstalk with the host immune system to increase what is known as a myeloid tone, or the inflammatory tone, and it raises your body's inflammation level. So people may know a little bit about immune therapy, they may know a lot about immune therapy, but certainly people have heard that inflammation is a bad thing, that it's a bad word. And that inflammatory state gets jacked up when you have certain key microorganisms that are adverse. And those bugs that appear to do that, particularly gram-negative rods, and those adverse microorganisms produce inflammatory cytokines such as IL-1 beta, CXL8, and certain things that essentially raise that inflammatory tone up. So when your immune therapy is trying to work, the bugs are actually working against that. So you're trying to do something, you're trying to get a drug, the drug is trying to turn on the immune system, and these bugs, they're just subverting that. And as it turns out, we can look at the genes and we can find the genes that do that, and certainly the genes that appear to be good are genes that essentially enhance your immune system. They alter the certain immune cells that are present that actually help the body's immune system work a little better, particularly what are known as MR1 restricted T cells and MATE cells. And then the bad genes that are found in the bad bugs, those genes essentially produce more LPS. And LPS essentially is lipopolysaccharide, and lipopolysaccharide increases that inflammatory tone. Now, we can also find bugs that are different in people who have side effects. So again, the people who get immune therapy, people who do well on immune therapy, don't do well on immune therapy, then the people who get immune therapy and do well or not do well from a side effect perspective. And when we look at the people who get side effects, we see that there's a very, very different gut microbiome signature amongst not just one side effect, meaning side effects versus no side effects, but you can literally fingerprint each side effect. You can say, you know what? This person has colitis. This person has a thyroid problem. This person has a neurological problem. And we have all these different side effect profiles. And the one side effect profile that's actually very, very interesting is a side effect known as arthritis. And the reason arthritis is very interesting is because the microorganism that tends to be associated with arthritis is this bug known as streptococcus. So again, streptococcus is not necessarily an organism that many people have heard of, but certainly in your 60s and 70s, streptococcus is the reason why we have what is known as strep fever. And streptococcus in the 50s and 60s used to be a common reason why people ended up with post-streptococcal infections that caused arthritic problems, but also cardiac problems. So strep is actually kind of well known in the medical space because people develop essentially post-streptococcal autoimmune problems that cause kidney, cardiac problems. And streptococcus is not found in the gut. It actually can't go into the gut. And the reason it can't go into the gut is because it's an oral commensal. It's only found in your mouth. And for it to go into the gut, it must mean that somebody is taking something that facilitates that, because it's killed by the low acid, the low pH or high acid environment of the stomach. And as it turned out, when we looked at that, we found that indeed to be the case, because streptococcus was only found in the people who took proton pump inhibitors. So another thing that's probably bad for you, that big guy that goes around on that ad on TV that says that you can buy Protonix at CVS, not a good idea. Protonix, bad. So finally, can we do something about this? Well, we talked a little bit, you heard a little bit about diet. People ask me all the time, can I do something about my gut microbiome? And the answer is a whole bunch of people are doing a whole bunch of things. So this is just a list of interventions that people are studying. There's everything from fecal transplants to consortia to dietary changes to monoclonal, what are known as monoclonal microbial candidates, where you just take a single bug and you try to give that to people. And I'll just give you a brief version of what people have looked at. So this is something we did, right? So this was a piece of work that was done here. And about 6 years ago, we started this experiment. It was very simple. It was just a proof of concept study in which we took people whose cancers had not responded to immunotherapy. Actually, the very first person that was treated was Dr. Kirkwood's patient. And we gave those patients immunotherapy, and we just gave them a different gut microbiome. We borrowed the gut microbiome from somebody who had done really well on immunotherapy, and we matched these people up by just checking the presence of certain key organisms to make sure they wouldn't reject that. And rather unsurprisingly, 9 people didn't benefit. But what was quite astounding was that 6 people who did not respond to Keytruda, did not respond to Opdivo, did not respond to Keytruda plus Opdivo, responded to Keytruda plus gut microbiome change. So all we did was we gave people a single fecal transplant, and 6 people started responding to Keytruda. So we're not here to say that you should, go on Amazon and start buying the reverse engineering kit that is designed to be able to do, fecal transplants at home. That's a terrible idea. Do not do that. We also don't necessarily think that you can start getting fecal transplants at the GI doctor's office. I certainly get calls about that. We think that certainly this proves that if you have a bad microbiome, that you're more likely not to respond. And maybe there's something we can do about it. We've been able to prove the reason why this works is because of certain inflammatory cells that are bad, myeloid cells, and certainly the people who do well on this tend to do well long term. In a more recent experiment, a bunch of guys in Montreal, actually across Canada, gave people fecal transplant derived from healthy donors, right? So these are young college kids. Those are the only people who are willing to poop for $50 a pop. And they took the poop from these young college kids and they gave them to people whose cancers were growing, who are candidates for Opdivo and Keytruda. And those people were given just Keytruda or Opdivo by themselves with 1 fecal transplant, right? So the standard response rate of Keytruda or Opdivo is about 35 to 40%. And in this study, they got the fecal transplant from 3 healthy donors. They treated 20 patients, and 65% of their patients, 13 out of 20, had radiographic shrinkage. And certainly those people have done very, very well for very long periods of time. This paper was just published. And a key aspect of that paper was something that we had shown about 4 years ago now, which was that the people who responded to a fecal transplant had what is known as engraftment, meaning the bugs that you got from somebody else survived. And then it's kind of like a transplant. It's any other transplant. You get a stem cell transplant. If your donor cells are not present in your bone marrow, they know your leukemia is coming back. They know the lymphoma is coming back. And much like this, if you get live bacteria from somebody else and the bacteria doesn't survive, it ain't working. So finally, what can we do about this? And what are people like me and other people doing about this right now? So a couple of things. One, we certainly think that immune therapy, to a large extent, is curative for a whole bunch of people. The problem is that we have a tail to the curve, and that fraction is only 25% for PD-1 monotherapy, 40% for PD-1 CTLA-4. We don't quite know what the durable tail of the curve is for PD-1 LAG-3, and certainly we're curing more people with TIL, and with the addition of some targeted therapies that is improving those odds, but it's still not everybody. And we're not going to be able to sleep at night until every single patient who has advanced disease is cured. And we think that clearly a large fraction of these people, 30 to 50%, are failing in the first 6 to 10 months, which is when we think the gut microbiome is probably exerting a greatest effect based on our data. And what we're trying to do, and what we think is happening, is that the beneficial microbes are needed to sustain immune therapy. But when you have a set of bad bugs, it's kind of like your wife's crazy cousin. The wedding doesn't go well because this person just disrupts everything. So what we're trying to do is we're trying to do proof of concept experiments to extend that. So certainly we've done this in refractory melanoma. That's already been done. There are now larger trials that are happening. In fact, the NCI is getting ready to think about a larger call for a national trial that we are involved with. And 2 independent experiments have shown this, both Israel and here. Israeli data is on the left, our data is on the right. And we've also now seen this from a Canadian group in an early study of a non-randomized phase 2 in advanced non-refractory melanoma. So what we are doing, actually, this trial is already open. We're hoping to accrue patients to it. And it's basically pembrolizumab and lenvatinib, which is a good combination in the refractory setting. 20-something% of people respond to this. And half of these people are going to be getting checkpoint inhibitor with the lenvatinib. The other half are going to be getting the same thing with the fecal transplant to try to get the response rate closer to 50%. And so this trial is open. What we're also doing is we're trying to develop an actionable biomarker, right? So we're looking at a whole bunch of people. And so the exact same people that get asked the question about whether or not you're taking high amounts of sweeteners or not also are asked to give us a stool sample. We've now profiled about 1,000 patients, and we're working on that data set right now. And we're looking at, again, the biomarker. And the whole idea is we want to see people who do well, right? So we know that 40% treated with one drug, 55% treated with two drugs are going to do well. And we think that the balance of the microbes, right, affects that probability. So if you're more good than bad, you tend to do well. And if that's the case, and we prove the biomarker works, and we have nothing more than we tell you that, you know what, you're already doing well. You don't need any help from us. But if we find that you have bugs that are bad, you need some help, right? And the question is, what is that intervention going to be? And we're working on that right now. We think it could be a gut microbiome transplant, a couple of new bugs. It could be a couple of key consortia, a couple of keystone consortia. Finally, side effects. Side effects are a huge aspect of what people who have treatment have to deal with, because it's one thing to put somebody into remission, which is good. It's another thing to put somebody into remission and have them live with long-term side effects that they are now living with for the rest of their lives. Because if the immune therapy is working and is producing a lifelong remission, certainly the side effects are also equally, unfortunately, durable. And we certainly are seeing a lot of these side effects more often, particularly because second-generation CTLA-4 inhibitors have been developed. So we've now got drugs that are producing remissions in colon cancer, microsatellite stable colon cancer. These are newer active agents from companies like Agenus. The problem is that when these drugs produce these effects in patients with colon cancer, and they work extraordinarily well in patients with melanoma, the reason they work better is because of some engineering that was done to the drug, but these drugs produce a bucket load more side effects. So we really think that side effects are an equally important part, because we not only want to cure every single patient, we also want to have every single patient not have to live with long-term side effects. So how do you do that? You have side effect trials. So what we have right now is a clinical trial where we're giving people a microbiome modulating agent to essentially eliminate side effects. And the idea is that if you have bad side effects, particularly skin or colorectal side effects, we'll enroll you in the trial, we'll fix your side effect. We think it's going to be about 40 to 50% effective in refractory patients, maybe 60 to 80% effective in patients who've not even had a biologic. So we think it's very effective in biologic refractory patients, but we're looking to study it earlier in order to try to prevent people from getting side effects. So the final thing, very simply, is what should you do? If you don't remember anything else, what should you eat or what should you not eat? So again, I'm not a nutritionist, so I can only tell you data that we or other people have generated, right? So we can say, what should you kind of sort of avoid, and what should you maybe eat more of? So I think clearly artificial sweeteners are bad, right? And we think it's bad because of data that we've generated, other people have generated, but you know, these things are not a natural product. You don't find them in strawberries. You don't find them in blueberries. You don't find them in a natural, they were made by DuPont. So I think that what we are concerned about is that the engineering behind these agents was just not designed to ensure that we know that it's going to be safe with everything else that people need. It was created as a substitute for sugar in the 1980s. The antibiotics that are given broad spectrum without concern for people being currently on immunotherapy is probably a bad idea. So the main thing is your oncologist is your best friend. And so if somebody's trying to give you something, maybe run it by your oncologist. What is good? Fiber is good, right? Do we think people are eating enough fiber? No, right? You know why? Because you know the USDA tells you to take 38 grams. How much are most of us eating? 15. So most of us are not even eating what we're supposed to eat, right? So if we ate a little bit more of what we're supposed to eat, we'd probably be thinner, we'd probably be healthier, we'd probably have less cholesterol, we'd probably not be fat, and we'd do better with immune therapy. So eat more fiber. Fermented foods might be good, and again, not beer, probably kimchi, sauerkraut, yogurt, things like that. The Mediterranean diet might be good, but again, you don't need to do this for your immune therapy. Mediterranean diet is good if you have cardiac disease, if you want to lose weight, if you want to have a lower glycemic index, if you want to have better outcomes from a BMI perspective. So many of these things are actually just commonsensical things that you should just do right now because you should already be doing them. So we don't, and we are certainly thinking about doing dietary trials to try to enable a better understanding of the space. And certainly my colleague, Dr. Zaror, who's the co-leader of the program with John, is engaged in some of these things. So these are my acknowledgments. We're very grateful to the patients that put up with our nonsense and answer our questions and give us stool specimens, because we literally, it's stool study, right? So we literally cannot do this research without you guys. So thank you for agreeing. Thank you for pooping in a cup. Thank you for not giving us your dog poop, because that's one thing we can tell. And many of these things are fascinating because this data, a lot of the data that I showed you is actually generated right here, right? So we're generating all of these things here with your help. So thank you.